So hi everyone. So I will just go over the very last point that I did last time with the plot, so that because I did a bit in a rushed way last time. So I had this overall rate that we said is the initial one. And I wrote down this K2 rate with the initial concentration of the enzyme times the initial concentration of the substrate, and then S0 plus K. And then basically I did 1 over R0, which is the reciprocal, and I wrote it in the following form. And then we had some slope here, which was K over K2 E0. And then it was multiplying the reciprocal of the concentration of the substrate, right? So basically I said that this is a linear function for 1 over R0 in this variable, 1 over S0. So if you were to plot this thing, basically you would get something like this. This is the axis of 1 over S0. This is 1 over R0. So the intercept would be this. And then basically if you had a bunch of data points, we said this would be the slope. So basically experimentally you could measure this by saying, okay, I know what the intercept should be is this, right? And then I can compute also the K from that, which is the slope. And we said that this is known as the Lineweaver Berg plot. Okay, so I will continue with the same thing before I move on to chapter 2. So now I will give you a quasi-steady approximation that we looked at last time. We already did this. And it has to do with the complex C given as S over S plus. So notice that now I'm using the lowercase letters. These were done from the non-dimensionalization last time, if you remember, right? Okay, so you will notice that if I use now the initial condition that I gave you last time, so what was the initial condition? If I go back to the capital letter S, it was S0, right? This was the initial concentration. But if you remember how we did the non-dimensionalization, I wrote that this is this essentially, right? So if I want to get the initial condition for the little variable S, this would be S of 0 is equal to 1, right? And what was the initial complex concentration, if you remember? 0, excellent, exactly. So I had also this. So you will notice that if I plug in here S of 0 is equal to 1, I will get 1 over 1 plus K prime, which is non-zero, right? So what's going on there? So I will say here, this does not satisfy our initial conditions. Oops, sorry. And the reason for this is, so basically I will just write whatever I said. This is C of 0 now is given by 1 plus K prime, which is non-zero. And the reason for this is because there is a transient region, or you can say here a rapid transient. So what this means is that if I look at the time, it's of order epsilon. And during this process, the quasi-steady approximation does not hold. Does not hold. Okay, so if I know that the time behaves in that way, I can now do the following rescaling. We use the rescaling given by this. So now this tau is a new variable that I will substitute into my equations that I derived last time for the concentration C and S. We had two ODs, if you remember. So if I plug them into the ODs from last time, I will obtain the following. So this is just change of variables. You can show it very easily. And if you remember, we also had this lambda, which was just a combination of some of the constants for the rates of reaction. So why did I put here 0? Because basically this epsilon is going to 0, is some very small quantity. So this tells us that the SD tau is now going to 0, which implies that S goes to a constant. This is to leading order. So we have that S is S0, which is a constant. Okay, so this is the equation for the SD tau. If I write down the concentration C for the complex with this new variable tau, we will arrive at S minus S plus K prime C. And now, since I know that S is just a constant from here, I will have that this is S0 minus S0 plus K prime C. So this is what type of equation I have. I'm trying to solve for C here, since I already know what S is. So I have this, and C appears here. These are all constants. How can I solve this? Yeah, it's the integrating factor, right? So basically you can write this in the form dc d tau. Maybe if you see it in this format, it will be easier. Right? So this is a first order differential equation. You can solve it using the integrating factor. So I will say here we use the method of integrating factor. How many of you actually want me to do it? None of you. Okay, so I will just give it. So basically you just do this. And then when you plug in this as your condition, I guess, or I should say here C, you will end up with the following. So this is a function of tau. It's 1 over 1 plus K prime. And I have 1 minus. And here I have already substituted for S0, which is the same as S0, is equal to 1, which was our initial condition. Do you have any questions so far? Okay, great. So when you get this, what you should notice is that now if I let tau go to 0, what happens to this? Exactly, which is what I need, right? C of 0 should be 0. So I will say here C of 0 is now 0. And as you exit the boundary layer, which we said is, if you remember, it was this, then what would we have? Tau going to infinity. If tau goes to infinity, where do you tend? Where does C go? So this goes to infinity, but it's minus, so it's e2 minus infinity is 0, right? So the whole thing goes to that. And that's good because that's what we had originally, right? Remember that I wrote C of 0 is equal to that, which is not equal to 0. Okay? All right, so this is what we needed. Now I'll introduce a new idea. It's something called an inhibitor. So how did this enzyme reaction work in general? Yesterday, when we saw it for the first time, we saw that enzymes are catalysts, right? Basically, you have this big molecule, which is an enzyme, and then something called a substrate binds on it. And then there's this catalysis reaction that happens, and it breaks it into something else. So this is what I have. This is my enzyme. The enzyme basically has some active site. Just imagine it has something on it, right? Like a lock. And then a substrate that is somewhere in here basically comes, and if it binds on this active site, the whole process can happen, okay? It can also bump on it and not do anything. That's not good. So now what we have is something called a competitive inhibition, which means that you can have some other molecule which is similar to the substrate, and it tries to bind like into the enzyme instead. So I will just define it. These are other types of substances that will inhibit the catalysis process. reactions of an enzyme. And we can have different types of inhibitors. So you can have ones that are bad, ones that can help the whole process. Let's see why. So I will give you the first example. The first example will be called competitive inhibition. So this is when a substrate cannot bind if this inhibitor is bound to an enzyme. So as I said, this is when a molecule is similar in structure to the substrate that we want to break down, and it doesn't allow basically the substrate to bind on the active site. Okay, so what can we have here? So I can have the substrate, which is the good one, and then the enzyme, and then I can have some rate K1. There is also a reversible reaction K minus 1, and it gives us the substrate complex. This is the thing we were saying, it's a two-step process. Then I have a reaction rate of K2, and we said it gives you back the enzyme, it doesn't get broken in the reaction, plus the product that we want, right? But while you have this process, you also have the enzyme reacting with the inhibitor molecule. And again, there is a reversible reaction. This is a different rate now, which I have denoted in the notes by K3 and K minus 3. And during this process now, when the inhibitor is reacting with the enzyme, you get a complex again, but it's a complex inhibitor. So it's Ci. It's a different one. So I will skip the process because I know you're really good students. You know how to do the law of mass action now. We saw it already, how to write the system. It's not the point of this thing. So I will introduce now the other type of inhibition that you can have. So that's example number 2. So it's something called the allosteric inhibition. So this is a bit different to the one before. So basically, when you have multiple binding sites, not just one, like the one I drew up there, you can have that when other molecules are coming, some can bind on one side, they can bind on more than 2, and so on. And while they do this, they basically change the 3D structure or the 3D shape of the enzyme. So they can either improve the binding process with the substrate or make it worse. Okay, but they change in any case the reaction. So this is what the allosteric inhibition is. So let me write just down something so that you remember. So as well as the previous type of inhibition, so these two reactions, we can also have the inhibitor binding to the CS to make, so this one, a different product. So during this two-step process, I have that CS. It also acts as an enzyme now, and it will work as a catalyst here. It reacts with the inhibitor under some rate again. This is K minus 1. This is K1. And it gives me a complex for the inhibitor plus the substrate. Okay, so from this now, we can also have, as I said, the more than one binding site. So this is what I will call cooperative system. So this is when you have more than one binding site. And I will show you a schematic representation of this for 2 active sites. Okay, so I will do it schematically, so it will just be a diagram of the enzymes. Let me see if I can change the... Okay, so this is what you have. You have the substrate, the enzyme. So I will just use a big molecule here for the enzyme. It can have, when I draw these lines, they just mean active sites, okay? So it has 2 binding sites. And then there is a small molecule, which is the substrate. It will react, they will react together. They will give us some complex C1. And this means it has attached to the active site of the enzyme. Now, there is a reaction again with a rate K2. It gives E plus the product. So it gives you the enzyme back in its original form. But now it has broken it down into some other thing, which is the product. Okay, so this is one of the steps. Then I have, we said, the S can also act on the complex C1. So again, I have this thing. This now is the enzyme, but with the original substrate bound on it. They react under some rates. They give us C2. Now, C2 is the enzyme. It has the original substrate, and it also manages to have the second substrate bound on it. And then I have some 4th reaction rate. And then it's C1 now. So remember, C1 can also act as a catalyst, which means I have this back to its original form, plus some product, which is this. So this is a cooperative system. So again, if you use the law of mass action, yes?
Sorry, should the C1s have the other active sites on them, or do you just not bother?
Ah, yes. I should have done this. You're right. It doesn't disappear. Thanks. Okay, yes, so this is what we have. So if you want to write it again as a differential equation, you use the law of mass action. You will definitely get a question on that, so be prepared. And then when you do that, to not spend your time on that, you can basically solve the equations again. It's relatively simple. You can get the final form. This is K capital. I will define these constants for you so that you have them. You get that the overall reaction rate is dependent on the substrate concentration through this relationship, K2S plus S squared. And then here I have that K1 is equal to the sum of this, K1, and then finally K2. The other constant is K4 plus K negative 3 over K3. All right, so if you remember from last time, yes, tell me.
Are all the systems operating at the same time? So is the competitive and the allosteric, like, do they happen at the same time, or are the eyes different?
No, it's the same. Basically, the allosteric can also have a non-cooperative system, so it can also make it worse. So I'm giving you an example of it. So yeah, when it has these multiple active sites, if a different substrate, not the one you want, is attached on the active site, it can also make it like a worse system rather than a cooperative one. Okay, so when you have this, you can also say the following. So if you want to see the full derivation, by the way, it's on the typed lecture notes. And if you have any questions on that, you can ask me. Okay, so I want to do a special case with this specific example. If now the rate of binding of the first substrate molecule is small, So the first substrate molecule is this. So when I talk about the rate being small, I am referring to K1. So K1 going to 0. If this is small, but the rate of the second substrate, that means K3, is large, then I have K1 going to 0 and K3 going to infinity. But here we have K1, K3 finite. You will notice that, so what happens? If K3 goes to infinity, what happens to K2? It goes to 0. If K1 goes to 0, K1 is the dominant one, right? So you can see if you plug in those two things that I just mentioned, you're gonna have that the R reduces to basically K4 is 0. And then I have S times S, so it's S squared there. And then I get K1, K2 plus S squared. Now at the beginning, I think it was at the beginning of last lecture, what I did was to define this Hill equation. So if you look at the Hill equation and you compare it to what I have just written, basically you will see that the exponent is some specific value. So this was R0 S to the n. And then I had K to the n plus S to the n. This is what the Hill equation was. So this is just some combination of the reaction rates. This is the original reaction rate. S is the concentration of the substrate we said. So it's with exponent 2. So exponent 2 in the Hill equation. And this is chapter 1. Congratulations, you just finished it. So I will do now something very interesting. It's called a transmembrane ion transport. So this is chapter 2 in the lecture notes. The lecture notes also have a section on glycolysis. We will not cover it. You don't need to know it for the exam. So if you're just interested, read through it, but it's not something you will be tested on. So chapter 2. This is called ion, or rather transmembrane, I should say, transmembrane ion transport. Okay, so first I will give you some definitions that you will need to know what I'm talking about. And once we have the definitions written down, we will do some equations again. So in general, if you don't know, cells are made up of a lot of water. So cells, you can think of them as bags of water. But this water is not just water. You have many dissolved ions within it, right? You can have sodium, chloride, potassium, all these things. So these contain dissolved salts. So basically you can have, as I said, different ions, but you can also have substances like sodium chloride or potassium chloride, which then dissolve into ions that have also charges, right? So you can have basically, sodium has a positive charge, chloride a negative one, potassium is plus. And basically, if you think of the cell, right, there is a cell wall, and there are these ions inside and outside of this wall. Okay, so basically, when these processes are happening, you can have the movement of ions from the inside to the outside and so on. So this creates basically a potential. Okay, so let me write these things down. These ions exist both inside and outside the cell, creating a potential difference. Okay, and the walls are not, basically are permeable, which means that they have holes and they allow this movement. And you can also have pores that are basically channels or so-called gates. It will become clear what those mean in a second. So I'll say the cell walls are permeable. Ions are transported through the membrane. Does anyone know what the process of movement of water from one part of the cell to the other is called from biology or chemistry? It starts with O.
Osmosis.
Yes, nice. Yeah, so passing through pores called channels. Sorry, I'm doing a lot of, I'm writing an essay, but I'm finishing soon. So this is gates or channels. And then as you guys said, is osmosis. This is the process or the mechanism by which water is transported through the membrane of the cell. Transported through the cell membrane. Okay, I have 3 more things to write down. This is important so that you can refer back to it. One of them is called, someone mentioned diffusion. So it's carrier-mediated diffusion. We will focus on this today. This is essentially what it means is that you have a molecule. I will write in a very colloquial way. So it hitches a lift. This is how it's also written in your notes. By basically binding on one of the carriers to a carrier molecule, which is a lipid soluble, and then it basically is able to transport around and move let's say through the membrane. You can also have the following process. So let's imagine I have the enzyme, which we said is a big molecule. So this is my enzyme. And now just imagine that this is the cell wall. Okay, I will have the exterior side, we said, of the cell wall and the interior. So let me just denote this as the exterior. And this is my interior. And then, as we saw in the previous example, you can have an active site. And this active site can have a substrate bound on it, right? So during this process, basically what can happen, you can also have the rotation of this thing. So basically what it means is that the active site originally can be on the exterior side of the cell wall, but it can also rotate in a way and it will be in the inside after. There is a better schematic also in the typed lecture notes if you want to see it, but basically it does the following. And I will do an example with equations in a bit. So we have a carrier-mediated transport. So this is exactly what I drew. Basically, a molecule binds to a protein. That has an active site. That might be exposed on the outside. Or inside of the cell. And it can also rotate, but I will not write that. Okay, so now that we have set this up, I will do a particular example and show you how this is written as an enzyme reaction. Okay, so the first model that we will see for this is a model for carrier-mediated transport. Okay, so you have the following. You have a CI and a CE. These are going to be the states with the binding site. When I do this is with the binding or active site on the interior of the cell wall. Interior. These are going to be exactly the same, but on the exterior. Okay, and the reaction scheme we are going to have in this case is going to be the following. I'm going to have the substrate. It's again I because it's a substrate that is found on the interior of this cell wall. It's gonna act on this CI with a rate of K plus, is going to produce some PI. So it's a product on the interior. This is also a reversible reaction. I'm gonna have K minus. This is not done yet. So now this product can also break down and it can give you a product that is now on the interior. So this has to do with this rotation that we were talking about. So you can also get PE. I will denote it with K. K is also in the opposite direction. So here I'm assuming that the rate at which this is happening is the same in both directions, okay? And then this product now on the exterior of the cell wall, this will be now the reverse of this. So I'm gonna have K plus here, K minus, and this is gonna give me exactly this, but on the exterior of the cell. Okay, and it's very useful to see this schematically, so I will show you how it works. And you also have on top of this, that basically these states that I was saying before that exist on the interior and the exterior can also be exchanged between them, which is the picture I drew before. So you can also have, maybe I should write it over here. So I have CI, it gets converted to CE, and vice versa. And again, it happens at an equal rate. Okay, so let's write these two processes. I will denote this by 1. So what you have schematically is the following. This is my enzyme. It has some active site. So this is my cell wall, say, right? This is the cell wall. And then I have some substrate here, which is Si. This is Ci. So this is the exterior, we said, and this is the interior. Okay, so this can now bind on that, and it can give me a product in the interior, which is the P1 that we wrote here, right? So this reaction basically can now give, with a rate of K plus, the following. And this is Pi. But we also said that this can also be reversed and it can give me the product in the opposite direction, so in the exterior. And this happens with a rate of, let's see, K, I wrote there. So this is gonna be K. And then what I have is this instead. So we write this, I will write PE so that I don't forget. Okay, but what happens also? The P can also be converted to that. So I have that this happens with a rate of K minus. This is a reversible reaction with K plus. And it gives me the same picture, but the mirror image of that. So it's going to be instead now CE, but without the substrate on it, right? So it's gonna be SE here. Okay, and then I also have the other state over there. So this is 2. I also have that this thing can also invert on its own without the substrate. So it can also give me, if I have CI, this can change with the rate of K into the following instead. Okay, so now to write down these equations, I will denote these different steps by different letters so that we keep track of them. So I will name these the reaction rate on the interior, so it's Ri. I will have also, this is just F, doesn't stand for anything, it's just a letter. Then I'm gonna use this, the reaction on the exterior. And then this is some process G. Okay, so for all these steps here, we can write down some equations, right, from the law of mass action, and we will be able to solve for this. Okay, so using the law of mass action, basically what we will have is I will write down an equation for all these, and then I will write it as a system for an ODE. So for now, it won't involve any derivatives, okay? So notice that I have the SI reacting with the CI under this rate, but during this process, you also have the reversible reaction of the PI being broken down into those things. So this means that I can write basically for this K plus. K plus is this reaction, so we said it's the product of the two concentrations. So I'm gonna write CI SI here. And then since the other one is in the opposite direction, I'm gonna have K minus and then PI, right? And here I didn't tell you what I'm looking at. Am I looking at S, the derivative of S, and so on? I'm doing it generically, and then I will write it into a differential equation. Okay, so let's do the F together. So the F is what? Does anyone want to tell me? The F1 is simple. Just do the forward first. It's gonna be K P1, right? Or PI rather. And then minus K PE. What else do I have? RE. So RE is this process, so I'm gonna write down K minus, and I have this. And then final in G is just KCI minus KCE. Okay, so when I'm writing down these equations now, I'm gonna make another assumption. So I'm gonna make an assumption that I don't want my final steady state to go to zero flux, meaning that I don't have any substrates coming in or out of my cell. So we'll just assume some initial flux of substrate that is moving around of J. It's just a constant, so that you can tend to that at long times, okay? Can I cover this board? Have you written? Okay. Okay, so now finally suppose that the substrate is being supplied from the outside at a rate that is constant, a constant J. And I will also assume that you have this flux of substrates that is being taken away from the interior with exactly the same rate. So it's gonna be minus J basically. So we have that this is being taken away from the interior at the same rate. Okay, and this is, as I said, to avoid a steady state where the flux is zero. All right, so now what I want to do is write down the equations. So how many things did we have? We had, sorry to keep moving it, we have a substrate in the interior, we have the complex in the interior. So I have two things, the product in the interior, the product in the exterior. So how many in total? 6, right? 6 things. So I would write 6 ODs here. So the first one that I want is d SI dt. So this will tell me how the rate of change of the substrate I is dependent on the other components. So I told you that I have a constant rate that you're taking away the substrate from the interior with J, right? So I should write minus J there, right? It's reducing the amount of J in the interior. And now you also see that reaction Ri is that decreasing? So should I include minus Ri or plus Ri for ds? What do you think? Minus, exactly, right? Because when it's reacting with the Ci, it's decreasing its size. Yeah, very nice. So it's minus Ri. Okay, so you get the idea, so I can now write the rest. So the exterior, similarly, can be written, now I'm adding the constant flux, so it's gonna be J plus Re. The product is positive Ri minus F. And then you have another 3. I'm gonna denote them by numbers because I will use it. I also have the product on the exterior. This is gonna be F minus the reaction Re, dCi dt. This is minus G minus Ri. And finally, I have dCe dt. This is G plus Re. So this is the 4th, 5th, and 6th equations. Any questions so far? Okay. I'm either explaining it super well or you're all amazing at physiology. I guess both. Okay. So now if I don't give you the J and it's an unknown, right? If J is an unknown, I have how many equations for how many unknowns? I have 7 unknowns, right? And then 6 equations. We have 6 equations for 7 unknowns. But notice that you can do the following. So we can say, I will write here now. So if J is unknown, I don't know, I wrote this, sorry. So we are gonna add the following equations. I'm gonna take 3, 4, 5, and 6. And you're gonna see that if I add all those equations, so what is 3? 3 is the product on the interior, the product on the exterior, and then the complexes in the inside and the outside of the cell. And you're gonna be now seeing very clearly that this is just 0. So this means that this thing in the parentheses is basically a constant. And I'm going to do another addition here. I'm also going to add 1 now, 2, 3, and 4. So 3 and 4 are basically involving the products in the interior and exterior. So when you do this, you also get that now the substrates on the inside and outside plus the products are 0, which implies that, so if you want to think about this intuitively, what this is telling us is that the concentration of the complexes and the substrate is conserved during the process. So this is the conservation of complex, or the carrier in this case. And this is the conservation of the substrate. Okay. So how many minutes do we have left? 5 minutes. Nice, I can cover a few things. We can ignore this, I don't know what it is. I guess it's from the previous lecture. So basically, once you have written these things down, you can also solve your system. So one can solve the 1 up to 6 equations that we have written down, and you will see that basically you get a bunch of constants multiplying the carrier on the exterior. And then I have the difference between the substrate on the outside of the cell wall minus the substrate on the inside, some constant, Km plus Se. And then I have some constant again subtracted. So let me define these things. So these rates are essentially again additions of the individual rates. So it's going to be K minus plus K plus divided by K plus. And I also have the K and D. So K and D in this case is just the ratio between K and K plus. So K, remember, is this rotation that we were talking about before. All right, so what does this tell us? What did we just find? Basically, when you solve it, you find an equation for the flux inside and outside of the cell. So from last time, so recall, I don't expect you to remember exactly the form, but it will be good at some point to know it. This thing, the Michaelis-Menten law that we saw last time, it was given by this formula. So I had the rate of change of the substrate concentration was minus lambda s divided by this. So basically this is, I know it looks very different when you just look at it, but basically you can think about the flux as a rate of change of the substrate. And then you can see this lambda is just a combination basically of those constant rates. And then I have S is the substrate, is basically this, you see. And then you also have an S in the denominator, which is this S. And then the K is just some other combination, so it's kind of similar. Okay, so this, so that you know what it means, and it's not just an equation, this tells us the flux of ions that are being transported across the cell membrane in a steady state. Okay, so the next thing we will do is again on carrier-mediated transport, but it's an active transport. So active meaning that I will need some energy to do the process. It doesn't just go down a gradient, it will try to go against the gradient. So for that you will need some energy, right? So we will see how that works in this case. So we'll work with potentials for this section. So it's an active transport. And this has a name. This is the sodium potassium pump. So I will first say what a pump is in the context of these cells. So these exchange one ion to another. So for example, you could have that you originally had some sodium ions, and then these are being changed for potassium ones, or you could think of Na plus with a calcium ion. And schematically, the way you can think about it is like I have these enzymes or carrier molecules. And then you're switching from like sodium, basically, you can think about having like calcium ions instead. So this is a pump. Okay, so what does it mean in terms of active transport? So as I said, you need to use some energy for this action. So the previous thing we saw, the carrier-mediated transport, moves ions down chemical gradients. To move against, this is important, a chemical gradient, we need energy. So this is why it's called an active mechanism. So in our case today, I guess I don't have time for today, but the next time what I will do is the sodium-potassium pump. And for that, you will need to know 3 different potentials, which we will first define, and then we will see again how you write the model for it. Okay, thanks for your attention today.